Hi, I'm Guna Mathivanan. Welcome to Simply Current Affairs. So today we will be discussing our 12th Current Affairs Hitlist topic. Friends, as you all know, we have started our exclusive Current Affairs program 75x75 and Economy full course targeting Prelims 2026. In the Current Affairs program, I will be completing 1000 plus topics along with static linkage wherever possible, and each class will be followed by a test based on that day's topic. To know more about this course, you can click the link given in the description, or you can visit www.simplycurrentaffairs.com, or you can WhatsApp to the number given in this video. Right. So today's topic is regarding genome-edited rice. See, if you haven't watched my previous Hitlist topic where I explained the difference between gene and DNA, so kindly watch that, because this topic you can understand it well when you have a clarity on what is the difference between DNA and gene. Okay, because I'm going to explain something called as genome. In this topic we will be discussing about gene editing, genetic modification, okay, and why this topic was in news for a significant reason. For the first time in the world, India has done something, so that we are going to learn in this topic. Okay, so previous class what we discussed, every cell has nucleus, and each nucleus has 23 pairs of chromosomes or 46 chromosomes, and each chromosome is nothing but compressed DNA. Okay, and specific segments of DNA, some important segments of DNA, that is called as gene. Okay, this I explained in the previous class. Now, what is genome? See, in the last video only I mentioned that on average an adult human being will be having around 30 trillion cells. Will be having 30 trillion cells. In all the 30 trillion cells, you will have nucleus. All the nucleus will have 23 pairs of chromosomes. All those 23 pairs or 46 chromosomes are completely identical, same only. So 30 trillion similar copies of cell. Okay. So in this 30 trillion, let us take one cell. Let us take one cell. So from this one cell, in the nucleus, you will be having 46 chromosomes. You will be having 46 chromosomes. So let us take all these 46 chromosomes. Okay. So we are taking one cell. So in this one cell, from the nucleus, let us take all the 46 chromosomes. Or 23 pairs of chromosomes. Okay, this 46 chromosomes is nothing but this 46 DNA. So we have taken all this 46 DNA. Plus, each cell will have something called as mitochondria. We refer it as powerhouse of the cell. It supplies a lot of energy. Now, inside this mitochondria also, you will have DNA. Inside this mitochondria, you will have some DNA. The main is 46 DNA is in the nucleus. So this DNA plus the mitochondrial DNA. Okay. So this DNA plus the 46 regular DNA. This put together, we refer it as genome. We refer it as genome. So genome is a complete set of DNA from one human cell. If you want to study a species, you want to study about any species, you come across a new species. Let's say, for example, you come across coronavirus. Now, you take the genome of this virus. So, in this virus is also a genetic material. This virus also contains, generally virus is made up of DNA or RNA. Corona is made up of RNA virus. So, you take all the DNA from the coronavirus, then that becomes genome. That means from the one cell I am referring to. So any new species, you come across a new fish species. Okay, new fish, you come across a new fish species. If you want to study about that fish, you take one cell. From that cell, you take all the DNAs out. That complete set of DNA is referred as genome. Okay, just taking and putting it in a box is not sufficient. We should know the sequence. we should know the genome sequence. We should know the genome sequence. See, in the last class only I was highlighting. Look at this image. In the last class I was mentioning that this DNA is nothing but it's a polymer. It's chain of repeating units. What will be repeating? I said nucleotide will be repeating. Forget about nucleotide and all. This DNA contains information carrying molecules. DNA contains information carrying molecules. I was mentioning about adenine, thymine, guanine, cytosine. Okay, so if you take the DNA, I was mentioning that all this all these information carrying molecules, they will be available in pairs. They will be available in this pairs. Now the order of this pairs, A, T, G, C, G, C, whatever, this order of this information carrying molecules, that is called a sequence. Sequence of one DNA, you will simply refer it as DNA sequence. Sequence of the entire genome. Sequence of the entire genome, we will refer it as genome sequencing. We refer it as genome sequencing. Okay. So I hope now you got an idea about what is genome. So in the previous class I explained human cell, nucleus, chromosome, DNA, what is this ATCG, what is this gene. Till this we discussed in the last class. So here genome edited, here I am explaining about genome. So genome is the entire set of DNA instructions found in a cell. In humans, genome consists of 23 pairs of chromosomes, right, as well as small chromosomes, there is small DNA within the mitochondria. So genome contains all the information needed for an individual to develop and function. When I say genome sequencing, finding out all the DNA sequence inside the genome, that is called as genome sequencing. So now we will try to understand what is this genetic modification. What is this genetic modification? We come across this term, right, GM crops, genetically modified crops. We come across this. So what is genetic modification? Now let us take a DNA. Okay. So these ladder like something which I am drawing, right, that refers to the base pairs. Now I said this is DNA. Some important segment of DNA which contains most important information. That important segment, okay, this part, let's assume this part, that is called as gene. See, in one genome, you will find approximately 20 to 25,000 genes. These are the most important parts of DNA. So what DNA does? DNA contains information. Now, based on this information, every cell will perform an action. Okay, so that information is very important. So DNA is like a giant book which contains a lot of information. In a book, not every page is important. Some selective pages are very, very important, that is having the actual formulas, etc. So imagine a giant book is DNA and specific insertion, specific pages, that is gene, important pages, that is gene. It is said that there is a particular gene in our body. If that gene is not functioning well, that can lead to 10,000 different diseases. One gene, okay, one of the gene, not any gene, one of all the 25,000 gene, one specific gene that is not performing their action well, then that will lead to more than 10,000 different diseases. That malfunctioning of that particular one gene could be the cause of 10,000 different diseases. Imagine. Now, what is genetic modification? So what I am doing is, I am going to remove, so I am removing this gene. okay. I am removing this gene. From this DNA, I am removing this gene. So now what I do, I come with a new DNA. So different species. So from different species, from a bacteria, assume from a bacteria, I took a bacteria DNA. So from this bacteria DNA, what I do, again, I am removing a specific DNA from here. I mean, specific gene from here. I am removing this gene. So this is a bacterial gene. This is the gene of bacteria. Okay, this is gene of bacteria. I am removing this and I am connecting this here. So I am connecting with the regular DNA. So what I did, I took a foreign gene. From a different species. I am linking that with the human DNA, assume. Need not be human DNA, it can be any DNA. I am linking that. So I am bringing a foreign gene and I am linking it with the regular DNA. This is called as a genetic modification. So if you take this BT cotton, now assume this is cotton DNA. Now assume this is a BT, Bacillus thuringiensis, that is a soil bacterium. So I took a bacteria's DNA and from that I removed a specific gene. Now this specific gene, I am inserting it in the cotton DNA and I am making the cotton DNA more powerful. So actually with respect to cotton, what is the problem? There is a worm called as bollworm. Now farmers are suffering because of this bollworm. This bollworm will come and attack, will destroy all the cotton plants. Okay, so now this bacteria has a specific capacity to protect the cotton from the bollworm attack. So what we did, from the bacteria, we took that specific gene which can make the cotton powerful. Specific gene we have removed and we are attaching it with the cotton DNA, and now we have created a genetically modified crop, that is BT cotton. So when I say genetic modification, the most important factor to be remembered is we are taking a foreign gene and we are inserting it to the regular DNA. But when I say genetic editing, genetic editing, gene editing, okay, when I say that, here let us take the regular DNA, okay, now a specific gene. So what we will do is, we are not going to remove it and all, we are not going to introduce any foreign gene. Rather what we will do is, we are going to do some edit within. Okay, this particular gene, so we have technology where we will slightly do some changes in the gene. Remember, we are not going to bring any foreign gene. Rather, in the same DNA, same gene only, I will be doing some editing. Okay, we say it as suppression of this gene or overexpression of the gene. So let's not bring new terms here. So we will do some kind of editing. That is, we will make the gene to perform more, or we might reduce the performance of the gene. This is called as suppression and overexpression. This we do, that is called as genetic editing. The difference between genetic modification and genetic editing is, in genetic modification, we will introduce a foreign gene into the regular species. In gene editing, we will not bring any foreign genes and all. From the same DNA, from the same gene, we will try to do some tweaking. Okay, so broadly there are three techniques. One we refer it as site-directed nuclease 1 technique, site-directed nuclease 2 technique, site-directed nuclease 3. The three techniques. In this site-directed nuclease 1 and 2, this refers to gene editing technique. This SDN3 refers to genetic modification. Okay, sir, what is a site-directed nuclease? That much technicalities UPSC will not ask. We are not biotechnology students, but just keep this SDN term in mind. SDN 1-2 means gene editing technology. SDN 3 means gene modification technology. Okay, one more thing which you have to remember. What is that? That is called as CRISPR-Cas. CRISPR-Cas or CRISPR-Cas9. See, we will discuss about this CRISPR in some other video. As of now, to know about CRISPR, this is an important tool for gene editing and modification. This is an important tool for gene editing and genetic modification. This is nothing but a kind of scissor, molecular scissor. The scissor is used to cut the genes, stitch the genes, modify the genes. So, remember two things. SDN1, SDN2 refers to editing, SDN3 refers to modification. Okay, these editing and modification uses a tool, uses a scissor to do the cutting and editing, that is called as CRISPR-Cas9. You understood the basics now. Now we will come to the topic. See, for the first time in the world, India has developed 2 genetic edited rice. So we will refer it as genome editing. Okay, we will refer it as genome edited rice. Genome is nothing but complete set of DNA from one cell is genome. So when I say genome edited, that actually means genetics has been edited. Okay, so India has become the first country to develop genome edited rice varieties. DRR then 100 Kamala and Pusa DST Rice 1 both have been developed by ICAR, Indian Council of Agricultural Research Institutions. Right? So which technology was used? As I said, scientists have used SDN1 and SDN2 genome editing. So this technique has been in use since 2001. For example, tomatoes were developed using this editing technique, fish varieties were produced, soya bean variety was produced, but for the first time, rice has been produced. Okay, India has done that. India has achieved that. Okay, remember this genetic editing or genome editing is not genetic modification, because in genetic modification, foreign gene will come. In genome editing or genetic editing, foreign gene will not come. So what is the significance? Now what we have done by editing the rice varieties, we have made the rice variety more stronger, more climate resilient. Now the new varieties of rice will be resilient to the climate change. It can withstand higher temperature, it can withstand salinity, alkalinity. Apart from that, it can improve the productivity, it can improve the yield by 30%. So this is going to improve our food security. When the yield is going to increase, that is going to improve the farmer's income. When these are resilient to the climate change, then that is going to help the farmers again, right? So all these are significance of what we have achieved. So this is a very important topic from our exam point of view, because first time this has been developed. So I have explained what is genome, what is genome editing. So precisely we will alter the DNA of an organism by inserting, deleting, modifying specific DNA sequence. The main difference between genetic editing and genetic modification. In genetic modification, so we are going to bring foreign genes. Okay, but if it is gene editing, there will be no foreign gene. That is the main difference between these two. Right? CRISPR-Cas9 is the tool which is being used for doing that cutting, editing, everything. Right, friends? That's all about this topic. I hope today's topic was interesting. So please connect this topic with the previous topic, only then you will have complete clarity. Right, I will see you tomorrow with another interesting Hitlist topic. Till then, bye. Take care from Guna Mathivanan. Before that, you create a multiple choice question based on the content which we have made in the comment section. Okay, so that it will be useful for others. It will also enhance your understanding capacity. Right, I will see you tomorrow with another topic. Till then, bye. Take care from Guna Mathivanan.